Morning class, how's it going? It's okay?
Good.
Good, nice.
How about you?
Things are going well for me. This is the most positive answer I've heard for a while. Most people are like, I'm surviving. I'm glad you are, but I'm also glad people are doing okay. Okay, so several things today. I'm glad you're all here. Um, there's a test on Friday. You know this. I will keep reminding you because I will feel really bad if you don't take it. But then they're like, rules about whether or not I can let you retake it, which usually is no. So take it. Also, your lab this week is the exam. Take that as well, right? Yeah, I know. This is what I'm here for. Also, tutoring's available if you're interested in it. Brief story, I don't know if you care, but I'm going to tell you anyways because I like stories. In undergrad, I took this Gen Chem class, right? And I was like, I'm going to be this bestest ever student. And I remember studying, I was reading my book, and reading it and thinking, I have no idea what's going on. Like I'm happy to read it, but I don't even know where I'm confused. I know I'm confused, but I don't know how I'm confused. And I was gonna go to the tutoring room. They had a whole room with tutoring stuff. I was going there, I was like, I don't know if I want to do this because I don't want to be dumb, and only the dumb kids are going. That's what I was thinking, right? Shut up, I'm like, okay. You get this figured out. Shut up, and realize the kids who were, my fellow students who were there were the bright kids. They're the ones who were asking questions and knew what was going on, and they were going to tutoring because they were figuring out how it actually worked, and I was like, imagine that. The people who study and think about stuff are the people who are going to tutoring, and this is great. It made my life so much better, and I actually finally understood what the professor was doing. She would draw these things on the board, and I had no idea what she meant, but then I did. Tutoring. Highly recommend. Also, there is a bio retreat. So if you're a bio major or minor, this is not really that optional. If you're anything else, you're welcome to come, but you're not at all expected to. There's a bio retreat. It's going to be on October 6th via Zoom. What it is you have these life plans and goals, right? Yeah. You're like, this is step one on a series of projects. We're going to help you make your 4-year plan. So we're not going to tell you what you have to do with your life, but we're going to tell you about when classes are offered, the rotations, ways it works, makes a lot of sense if you're going pre-med, things that make a lot of sense to try if you're going pre-vet, things to do if you're planning on some sort of conservation career, right? Different options. There'll be upper class men, women, upper class people there who will help. You can kind of get this plan planned out, all right? Then you don't end up junior year, that's hard, 2 words to say together, realizing, oh no, I've never taken physics or whatever, okay? All's well, makes a lot of sense. Okay, today we're going to talk about membranes. They're great. So, sorry, questions about the administrative stuff? Things make sense? All right. Sorry. Okay, the plasma membrane is this boundary, right, that separates cell and not the cell. Yeah? Reminds me what it's made out of? Phospholipids, right? This phospholipid bilayer, it's beautiful. We're going to mostly be talking about that membrane on the outside, but all those membrane components of the cell have very, very similar membranes, okay? So the phospholipid bilayer structure, the rules are basically the same, okay? It's selectively permeable, meaning some stuff crosses more easily than other stuff. And we're spending most of the day on this semi-permeable concept. So your book calls it a fluid mosaic, which is just a way of saying this membrane isn't rigid. It's still fluid. Things can move, It can move laterally within it. And it's a mosaic because it's not all one thing. It's several different parts. Phospholipids are the big one. Far and away. So many phospholipids. There's also going to be proteins. So phospholipids are going to be this kind of thing here, right? A whole bunch of little phospholipids, phosphate part, lipid part. This phospholipid bilayer makes a nice, stable layer between what are called aqueous compartments. So there's water on the outside, water on the other side. Phospholipid bilayer has this like boundary there, right? You are absolutely not expecting one little phospholipid to just like decide to leave, like whee, and pop off. Okay? You're not expecting that. So you have this. You then have this exciting picture. You've got all these proteins stuck in it. That's good. You've got some cholesterol here. You've got some carbohydrates stuck on there. You've got some other things going on too to make it more exciting. Mostly phospholipids, a bunch of other things too. All right? Now, all these phospholipids are basically the same thing, right? And you're not expecting them to like pop off, but they can drift. So you can imagine like, I have to think of these things during class, and I didn't realize I should bring things. I'm going to bring food coloring on Wednesday. But you can imagine like if you have a plate full of marbles, I don't know why you have a plate full of marbles, but you have a plate full of marbles, right? You could, have you ever done this? Like you could roll your hand along and move the marbles, but still keep them all in the same plane, right? They all still are there on the same level, but they can move amongst themselves, right? We're okay with this picture? The membrane can do the same thing. Phospholipids can move laterally within the bilayer. They can go sideways. They don't come out, except in very specific ways we'll talk about later. They don't pop out, and they don't pop in, but they can drift laterally. All right? Most of the lipids that are there in the phospholipid bilayer can do that. Some of the proteins can do that. Some of the proteins are anchored in place, but a lot of them can move as well. So then, if you have something like this, just to illustrate the point, if you have some sort of two cells, a mouse cell, a human cell, you have all these nice little mice protein over here, human proteins here in purple. If you smush the two cells together, I don't know why, you're bored, you smush them together right next to each other. After a while, things are going to drift, right? Not because anything's pushing it, just because things drift. After a while, are you going to still have that picture where the pink is all on one side, purple's all on the other side? You're shaking your head. You're right. Why not? You don't have to answer if you want to, but you're right. Anybody else want to take explain to the class? Yeah. It just moves. It does the thing, yeah. So after now you come back and you're like, yeah, things just all squiggled around, okay? Again, none of these proteins have like a motivation, alright? It's just things wiggle. Alright? We're good? So you've got this, we've got this cell that has this membrane that's somewhat fluid, is the term that's used. Somewhat movable, alright? It's not completely mush, but it's not stiff either. And this is the reason why. Briefly, before I talk about this slide, the most missed question on a quiz 2 weeks ago was one about a fat. It had a fat there, and it was drawn. I think maybe just the structure wasn't something people were used to seeing, but it had a fat drawn, and the question was something along the lines of, what is this? And it was like, is it carbohydrate? Is it a saturated fat, unsaturated fat? protein, who knows, right? It was a fat, but how can you tell a saturated fat from an unsaturated fat? Yeah, if you've got a carbon double bond, you know it can't be saturated. Because saturated fats have every single hydrogen they could possibly have. If you have a carbon double bond, instead of having the space for carbon to bond to that hydrogen, you've stolen that and had that bond with carbon, and now you don't have as many hydrogens. Do people want to see that on the board, or are we okay? On the board? Yeah. So, we're going to ignore most of the fat, because the fat's going to have that like alcohol group. We're just going to look at the carbon part of fat, the carbon hydrogen part, okay? How long should this be? 2, 3, 4 carbons long? 4 carbons? Okay, 4 carbons? Okay, so 4 carbons. We have this one. Just a sec. Sorry, I need to make sure things are viewable. This is like impossible to see. Just a sec. There we go. Okay. So I just make it so online people can see better. Okay, 4 carbons here. Great. Four carbons there. Cool, cool. In this one, if I have a double bond here, okay, I'm going to just put one double bond in. Here doesn't. This one I'm going to start putting hydrogens around. How many hydrogens go here? 3. Nice. Here. 2. 2. It's the same thing. And then 3 again. Great. Okay. This one over here, how many hydrogens around here? 3. How many hydrogens here? Only 1, right? Here? 1. Here's 3 again. Okay. So this thing is saturated with hydrogen. Every possible spot for hydrogen has a hydrogen, right? There's no way to put 4 carbons together and have them all connected to each other and have more hydrogens attached, right? To put enough, just full as it can. 4 carbons attached here, but because the carbons are double bonded here, we only get 1 hydrogen at each of these spots. So this is unsaturated. So unsaturated are going to have at least 1 double bond. Saturated aren't going to have those double bonds with carbon. Okay? This double bond makes it bend. When it bends, it's got a bend in it. And I'll show you a picture here. Can anybody think of some sort of fat or grease that's liquid at room temperature? Say what? Peanut oil, right? This stuff, it's grease, it's liquid, right? Sorry, it's fat. But at room temperature, it is super fluid, like really, really liquidy, right? Crisco, at room temperature, this stuff is solid, right? Like, it's not going anywhere. If anybody had to hazard a guess, is Crisco saturated or unsaturated fat? Saturated, yeah, you guys know, yeah. It's saturated, right? Mostly. So saturated fats, because they don't have the double bond, they're really straight, right? And they pack tightly. So something like if you had something like this right here, all these saturated tails packed together, this stuff's pretty stiff. It's fairly solid, all right? if you have something like peanut oil, where you have a bunch of unsaturated fats, they've got to bend. And that, it's harder to pack it tight because they stick out funny shapes. And so everything's more fluid, right? Now, cells, and specifically we're going to talk about animal cells, want some of each, right? They want to be stiff. They want to be at least somewhat structurally sound, right? They don't want to be complete mush. But they also don't want to be Crisco. They want to be so solid that nothing can wiggle, right? And again, I say want as if they're aware, but it's not a good way to have it be. So the solution for animal cells is this picture down here, where you have a bunch of saturated fats on these phospholipids. So things pack really nicely, right? Packs beautifully, but you disrupt that structure a little bit. So you put a few cholesterols there in the membrane. Now cholesterol is a lipid, right? Nod. Yes, excellent. Cholesterol is a lipid. It's happy to hang out with the other lipids, right? Not a problem. Happy to hang out there, right? It hangs out there, but it messes up the packing. Not terribly, like it's not complete goo now, but it's a little bit more fluid than it would have been. Everyone's comfortable with this? Okay. So that's the actual just phospholipid part so far. But you can stick stuff in this. You can stick proteins in this. So there can be a whole bunch of different types of proteins you stick in. And we have 3 types. And I'm going to draw a picture on the board because why not? All right. Back of the class, is the chalk viewable? Can you see it okay? It's fine? Okay. All right. We've got our phospholipid bilayer. This is kind of a lazy way to draw it. Oh, don't break, computer. Let's see. Yeah. Okay. Kind of a lazy way to draw it. This is outside the cell, inside the cell. Because of this, are these phospholipid heads here? Yeah, these are the heads, and the tails are pointing in. Right? We're okay with that? Okay, cool. There's legitimately no wrong answer here. Where would you like to put a protein on this thing? Just pick a spot, any spot. This spot right here?
Yes.
Beautiful. She wants the protein right here. She wants it to be attached to the membrane. I'm assuming you want it to be attached to the membrane. It's attached to the membrane, so it's holding on. But it's on the inside, right? It's doing some function on the inside of the cell, right? It's attached here. It's not going to go. It's there. We need it for whatever reason. We want it to do stuff on the inside. Cool. With that same logic, you can have one on the outside too, right? Be like, yeah, cool. We'll put one over here. It'll like be held on by the membrane, but it's mostly on the outside. I mean, actually, these are actually better. So far, so good. What's the other option? In the middle, right? You can have ones on the edges, or you can have one in the middle. So ones that are on the edges are called peripheral proteins because they're on the peripheries. They can be on the outside and the inside. They're bound to the surface, right? Oh, actually, this is a better way to draw it. Sorry. There we go. These are peripheral ones. This one's an integral protein, it's integrated into this membrane. Okay with that? And if you get all excited and you have it go through but stick out a lot, it's called a transmembrane protein. It's on both sides. Cool with that? Got this protein here. If you had to just take a guess, so there's definitely water on both sides, right? If you had to take a guess, do you think this part of the protein likes water? Yeah, right? Like, it's cool hanging out with water, this part? Yeah? What about this part? No, you're shaking your head. Why not? You're totally right. You're absolutely right. Just because of where it's at, right? Yeah. Um, if this thing was polar, it would like water. But if it was polar, it would not like being inside this fossil bilayer, hanging out the lipid tail. Anything that's like in this part, you expect that to be something hydrophobic, something that doesn't like playing with water very much. And so it hangs out nicely with the lipids. Does that make sense for us? Great. You're good. So that's some sort of picture like this, right? Where we've got, integral protein, peripheral protein. Question, is it on the edges or is it integrated into the membrane? Integral, right? Yeah. And all that stuff in the middle there, these ones are hydrophobic, and they're being held inside the membrane. Yeah? Beautiful. Come on, change. Okay. Proteins that you have stuffed in or next to the membrane can do a bunch of stuff. And we're going to see them all, briefly. So they can do things involved with transport. So they can basically make tunnels, right? They can let things go through. They can have these what are called carrier proteins, where they like let something come in and then spit it to the other side. This takes energy. This one just is a hole that lets things go through. You can have some of these ones on the edges are enzymes, right? They're going to speed up some sort of chemical reaction. Signaling is a huge part of this. So cells often respond to, say, the outside environment, right? And then change what they're doing based on the outside environment. So you need something that can interact with the outside. So you'll have a membrane protein that can notice the outside. So for instance, you might have a receptor that can notice insulin, right? So insulin comes along and it fits nicely into this protein. And this protein, basically it's like a key, once the key goes into lock, right? Once insulin goes onto the receptor, the cell's like, I should do stuff. The protein changes shape and sends a signal into the cell being like, oh, I have news, right? We're okay with this? Cells also often need to recognize other cells. So, for instance, with an immune system, if you had some sort of immune system cell whose job it was to notice foreign cells, something that's not part of you, you'd want them to know what cells were your cells, right? You want them to know, hey, this is me, don't attack it, right? Cells can recognize each other. So one of the ways they do that is by having these proteins, and usually there's some carbohydrates associated with it, that the cells can recognize. Being like, oh, hey, I know what you are. Yeah? Does that make sense? There's also, it looks similar, but the function's different. There's also intercellular joining, which is just basically saying one cell wants to hold on to the other cell, right? Don't want these cells to go floating off. They want to grab on. Most tissues will have some sort of version of this, where they'll hold us stick together. And then Another version that looks kind of similar but slightly different. Some of these proteins are going to hold the cytoskeleton on one side, and they're going to hold the extracellular matrix on the other side, so that the cytoskeleton doesn't move relative to the extracellular matrix, so it stays still-ish. They're still on jiggling. Functions that make sense? Basically, having proteins on the membrane can do lots of things, right? Because they can talk, because it can see outside, it can anchor things, it can connect things, right? And obviously, I realize they're all drawn as like bluish purple blobs. There are different types of proteins for these things, right? Not all membranes are identical. The phospholipid part is pretty constant. The proteins you put there is what's going to make it actually interesting. Okay? Cool. And then this next slide is just explaining how cells grab each other. We need to see this next picture here. We've got the ER over here, right? We have ER, we have some vesicles floating around, we have a Golgi, we have the plasma membrane of the cell itself, and then the outside of the cell, right? Okay. All this orange goo here is the cytoplasm. Yay, orange goo, okay? It's not really orange, but for this picture, it's orange. This orange stuff, right, is touching this orange side of the membrane, right? Anything that's in here in blue, on this blue side, that's outside of the cytoplasm, right? It's not touching the cytoplasm. So, from this little vesicle's perspective, anything here is outside of the cytoplasm. Yeah? Because it's not touching it. All this blue stuff is not touching, right? When you got the ER, when a vesicle buds off from the ER, you had this little vesicle. Anything that was on the cytoplasm side, still on the cytoplasm side, right? The orange is still on the outside. When it comes in and goes to the Golgi apparatus, same deal, right? Outside still outside, inside still inside. Do you see that? And then when this vesicle buds off and it goes and fuses and spits its stuff out to the extracellular side, it still has the side that was touching the cytoplasm is still touching the cytoplasm. Is everyone okay with this? This picture, it makes some intuitive sense, I hope. Okay. The fancy way to say this is that membrane faces are conserved. So faces are just the sides, and you don't flip them. So if something's touching cytosol, it's always touching cytosol. If something's not touching cytosol, it's never touching cytosol. It doesn't make any sense to, it doesn't happen, like, you don't have one of these vesicles that's inside out. Okay? Are we comfortable with that idea? The trick for some people is right here the blue is inside the vesicle, but here the blue is outside the cell, right? See, that's like way, but that's okay because this has always been outside of the cytoplasm proper, and it's still outside the cytoplasm. Everyone's with me there? Okay. I had a cell molecular, a cell molecular class that was like, hold on a minute. But with the picture, we're okay with this? And the proteins, the way they stick up or out, is different based on which face, on which side they're in. So these little membrane proteins that are supposed to be sticking out, notice right here, they're pointing into the vesicle, which makes perfect sense because they're gonna never be touching the cytosol, and when they're out here, they're still not touching the cytosol. Everyone's happy? You are a fast class, by the way. Great. So, deep breath. We're comfortable with kind of the basic structure of this stuff. Now that we have this stuff, this membrane, what does it do? It's selectively permeable. So, cells need to have stuff leave and they need to have stuff come in, right? This is like your city-state idea. You need to be able to have stuff coming in and out. You're not self-sufficient unto yourself. So plasma membranes are going to be selectively permeable. They're going to let some things through, but some things not. So I realize you probably know the answer to this, and this is good. How come people put screens on windows in their houses? Better question. How come you don't just leave the window closed all the time? Just keep it closed?
That is so you can let air in, but not the bugs.
Yeah, you want to let air in, but not bugs, right? Because if you have the window closed, you keep bugs out, but you also keep the air out. Yeah? If you have the window completely open but no screen, you let air in, but you also let bugs in. And whatever else. Possums, who knows, right? But with a screen, you can let air through, but keep the bugs out. Yeah? A screen is a selective barrier, meaning some things are selected, like, some things can go through, but other things can't. Everyone's with me there, conceptually? For the lipid bilayer, it's permeable to non-polar molecules. Small non-polar molecules are best. To something like O2, non-polar, can just diffuse right, can just squiggle its way through this, not a problem. Okay? Just can pass without, doesn't need any help. It's like air going through a screen, right? Cool. Things that are, say, charged or polar, so some sort of like ion or something that's very polar, something like water, doesn't cross very well. Okay? Doesn't cross very well because you got this lipid bilayer, right? Like something like water wanting to go across. Water is big. Water is polar. And for water to cross, it would have to have that big polar part in there in the middle of all those lipid tails. And the lipid tails are specifically organized the way they are because they're not wanting to hang out with water, right? Water is not squiggling across the membrane just by itself. All right? Similar deal with something that's charged. If you say have a sodium ion, if the sodium ion is surrounded by water, it feels pretty calm and stable, right? The water molecules can kind of support that charge. If that sodium ion has to be just by itself and I guess naked in the middle of the little in the middle of the lipid tails, it is not happy, right? You're not expecting ions to cross just by themselves. Yeah? But maybe I want to have water or sodium ions inside my cell. What do I do? Yeah, absolutely. I don't know if everybody heard what Ben said. You're going to put it inside something that lets it through, right? Right? So just because you have a screen on your window, it's not like, well, I guess nothing will ever come into my house, right? Like, you've got doors, right? Okay, so this is why we've got this cat door picture here. Our neighbors have a cat door, and our cat likes visiting them a lot, which is nice. Our neighbor's nice. Our neighbor's cat also wants to come into our house, but we do not have a cat door, so we're like, sorry, cat. Transport proteins are going to do just what Ben said. They're going to let hydrophilic substances cross the membrane. So they're going to be basically a door. Okay? They come in a couple of versions, and I'm going to go back and show you one because, well, no, I'll just talk about it. Some are called channel proteins. They're like this little guy here. There's just basically a tunnel. So then, some like iron, well, I guess you could have water go through, right? Because it's always hanging out with other water. It's never having to actually touch the lipids. Does everybody see that okay? You can also have slightly different ones. They're called carrier proteins. They're something like this, right? Where you have something coming in, you change shape and then spit it on the other side. Again, this thing never actually has to touch the lipids. Right? Does that make sense for us? Things can now enter or exit the cell, but they don't have to be physically in, I mean, they cross the phospholipid bilayer, but they don't have to interact with those hydrophobic lipids tails. Yeah? A really important channel protein in membranes is something called aquaporins. This porin idea has to do with pore, like a hole, like your pores, right? Looking at the aqua, any idea what it's letting move through? Water, yeah. Aquaporins are a big deal. So you've got these channel proteins, they're going to let water across. Fair enough? Concerns about this? There's a problem fundamental with cat doors, which is solved by cells. When you have a door or a hole in your house to let some things through, right? So the idea is, yeah, we want to make sure our cat can go in and out. Other cat-sized things can get through, right? This can be a problem, right? Do you have a story about this? No. Okay, some people have this facial expression like, yes, I will never forget the night of the raccoon or whatever, right? Okay, things can come through. Cells want to let big things through, but they don't want just random junk, okay? What cells do is they have something called specificity, or they can be selective. So a transport protein is specific for the substance it's moving. So if you have some sort of channel, some let a bunch through, but a lot of them are shaped in such a way that it only lets a specific shape of molecule through. So it might be just the right shape to let water through, but it's not going to let other molecules through, right? Or it might be one of those carrier proteins that, yeah, it'll let sodium go through, but it's not feeling potassium. Potassium doesn't fit right. Does that make sense? Okay? So just because you have a door in your cell's wall, just because you have a door in your wall doesn't mean you let everything through. Just because you have a channel protein in your cell membrane doesn't mean you have to let everything through. So it's still selectively permeable. All right. The last concept for today, we're going to start. We are not going to finish it until Wednesday, all right? It's worth noticing. Yeah, and it's diffusion. And I need to draw stuff on the board. All right. Seems like it goes very, very long ways. Okay. Stay with me here for a moment. Actually, let's make it a bit smaller. Anybody here ever babysit a bunch of toddlers? People's eyes go big, right? Let's say you're babysitting 9 toddlers, because why not, right? Just because you're tough like this, right? You got 9 toddlers, and the person who's watching them before you, for some terrible reason, filled all their sippy cups with Mountain Dew, right? Right? Okay. What are these kids gonna do? Right, okay, right? They're everywhere, okay? Might not have a lot of focused goals, but they'll move, right? They'll just do their thing, okay? So you've got this room, you've got these 9 kids, right? All over the place, okay? Cool, right? If suddenly you open a door or somehow make the room bigger, would you have to physically, take a kid and put them over here, or are they just going to spread out? Yeah? Even without any push from you? Yeah, how come? You're like, they just do, right? Is everyone okay with this? At the end, after a little while, they're still moving all over the place. After a little while, if you were to look, you'd expect toddlers spread out. Oh, I had 9 of them, right? About like that, right? They'd spread to fill the space. right? Once they spread to fill the space, do they all freeze in place? No, they're still going to move, right? Okay. You see where the dumb analogy is going? Molecules have, well, there's motion inherent in molecules, right? Other than at absolute zero, but that's a physics problem that's beyond us. If you've got molecules in something, say a solution, you've got dye molecules, you've got several of these nice little dye molecules. It doesn't have to be dye, it can be anything. They move. They move just because molecular motion makes them wiggle, right? Like, if you have a bucket of water and you drop a thing of food coloring on the top of it, and then you go do whatever it is you do and come back, that dye will have spread throughout the bucket, right? Yeah? These molecules, just with molecular motion, they wiggle around. And because it's fluid, there's a lot more ability to wiggle. Molecules in something solid wiggle too, but they're tied down more, right? But here, they can just move amongst each other, right? Does this molecule have some sort of deep, abiding personal quest to go over here? No, right? It's not self-aware. But it wouldn't surprise you if, over the course of time, this molecule ended up over there, right? We're okay with that. Okay? Kind of like we're okay with the toddler idea that they wiggle. If you've got these molecules and then suddenly there's a higher concentration of molecules on one side of something than the other side, they will spread out. They'll move to fill the space, right? And at the end of the day, you're expecting something like this. So far, so good? Great. That's the basic concept of diffusion. I have some official ways to describe it on the slides actually. But if you have the idea of little kids moving, kind of the same general concept. Okay, so diffusion is just describing the tendency for molecules to spread out to fill the available space. You don't have to put energy in to do this. You don't have to pay some little tiny microorganism to be moving these, okay? It doesn't cost anything. It just happens. Each molecule is moving randomly, but there can be a net movement. So net just means overall, like net income or something, right? So just because we have all these molecules on one side, and then in general there's movement to the other side to fill the space, each molecule itself gets to be moving randomly. Does that throw anybody off? We're comfortable with this? Comfortable with this? Yeah. And at equilibrium, you're expecting as many the concentration on one side to be the same as the concentration on the other side. Does everybody know what concentration means? It's basically molecule per unit space or per other molecules, right? Okay. So here's briefly a picture, and we will, there's a lot more, but we're going to stop with this picture and I have a question. We've got right here all these nice little molecules of dye. This membrane is permeable to the dye. The dye can cross the membrane. So this dye is going to squiggle, and you're going to end up at equilibrium. You're going to have equal concentrations on both sides. Yeah? Does it bother anybody this picture where right here there's net diffusion of orange across this way, but net diffusion of purple the other way? Does that bug anybody? No? Okay, you're okay that you can have net diffusion going in different directions because the concentrations are different on each side? Yeah? Great. I have a sneaky question then. So go ahead and take out your clicker things. I tried, so for reason there's nothing in A, if I made question A, B, C, but then hit shuffle, I don't know why it does, what it does. Okay, let's try this. And anybody who's, why is it not showing?
Yeah.
Oh, thank you. There we go, thank you, class. If I was missing one just at the end, just tell me who you are, because then I'll, did I double click it? Oh my goodness. It's not frozen, right? It's okay. Okay. Who, if I'm still missing you, just hold it up nice. There we go. Wait. There we go. Missing anybody? Great. Great. Oh, there we go. Is it trying to graph? Stay put because it helps me a little bit. Because I think there's going to be a discussion here. Ah. Okay. So nobody picked A, that's good. I can, this was made sneaky on purpose. At equilibrium. With our toddlers at equilibrium, right, when they're spread out across, right, when there's equal concentrations on both sides of the room, have all the toddlers frozen in place? No, okay. It's a really common misconception that at equilibrium, the molecules stop moving. Because they're like, yeah, there's like, it's equal. Just because it's equal, the molecules still move, okay? But there's no overall movement of molecules. There's no net movement of molecules, meaning there's not like a sudden migration of all these molecules from one side to the other. It's at equilibrium, you're expecting every time one of these little molecules waddles over here, it's not really waddling, you're expecting one from this side to go over here at about the same rate. Does that idea make sense? Before you're at equilibrium, you do have net movement. So right now, there's no dimolecules on this side, there's a bunch on this side. Before you reach equilibrium, you're expecting a net movement of molecules across that. But once you get to equilibrium, you don't have a net movement, but you do still have movement. Be comfortable with all that? Sweet. That's what we've got. Have a wonderful rest of your Monday. People who didn't have clickers, please tell me who you are.